So structure of HIV. So human immunodeficiency virus. So this belongs to a family called lentivirus. So what is the unique feature about this virus? These viruses can be seen only by electron microscope, not by a common microscope. Only by electron microscope these viruses can be seen. So these are very small in size. What is the size of it? This is almost 100 to 120 nanometer. What is called nanometer? 10 power minus 9. So the diameter of these viruses is about 100 to 120 nanometer. What is the shape of it? From the shape it is very clear. It's spherical. It's almost round in shape. So it's round in shape and the diameter is about 100 to 120 nanometer. Okay, and in center of this HIV you can see a dense core. Dense core means very thick, highly populated organelles are just present. So it's dense core. So this dense core again covered by a protective covering. There is an outer covering for the dense core that is made up of lipoprotein. So this envelope is made up of a substance called lipoprotein. So this lipoprotein only covers the inner dense core of this HIV. So what are the chemicals present inside this envelope? The envelope of this outer region of this HIV. The outer region of this HIV is made up of a knob-like structure, almost like a door knob. So these knob-like structures are called as a glycoprotein. So out of this glycoprotein, they got two different parts. One is a spike-like cavity, this rod, and one is a blue color round knob-like structure. So these two parts are just present. So in that, this round blue color round part is called glycoprotein 120. Whereas this yellow color knob-like structure is called as a glycoprotein 41. So first one is GP41. What is GP41? This elongated spike-like nature is called GP41. This blue color round-like structure is called GP120. This color difference given only for each identification. This is not the exact color of HIV. Okay. So at the core, at the center of this HIV, it has got a two single-strand RNA molecule. So two single-strand RNA means it's commonly called DNA, but here DNA is not combined together. They just form as a two single-strand RNA molecule, and also got a very important chemical called reverse transcriptase. So this chemical is a unique. That is why it's always been difficult for finding vaccines or medicines for this virus. So as a result of the reverse transcriptase, keep on changing their genetic material. They either change this RNA into DNA and DNA into RNA. And along with that, it also got enzymes like protease, ribonuclease, everything are just present inside this. And the core is covered by a capsid made up of protein. So the inner region is made up of a capsid called protein. So there is also, it's followed by another layer of matrix protein. So inside there is another layer that is called matrix protein. So that is about the structure of HIV. Then the next topic is how does HIV transmit from one person to another? So what is the method of transmission of this HIV? So the HIV is usually located inside the cell, especially inside the macrophage, that is your WBC, specifically T helper cells. So inside this macrophage only this HIV virus is present. Once it just comes out of this HIV inside the cell, outside the cell, it can just be survive only up to 6 hours. More than 6 hours, this virus will just die. Inside the cell, they can just present about 1.5 days. When they just go inside the cell, they can be inactivate but alive for about 1.5 days. But once they just comes out of the cell, within 6 hours, it should just go and invade the other cell. If not, this particular HIV virus will just die. So during this period of infection, so what will just happen? So what are the various routes of HIV transmission? You already know this. The various methods of HIV transmission is unsafe sexual contact, blood contaminated needles, organ transplantation, blood transfusion, vertical transmission from the HIV infected mother to the child. So from your mother to a child through placenta also they just transmit. So after getting transmitted into that particular person, after getting transmitted, what is happening? It just goes inside this T helper cell, if not this macrophages, where the RNA genomes are just been altered. It just changes into viral DNA. It just goes inside all this mRNA, everything has just been altered. This entire macrophage DNA has been altered. It just changed into a viral DNA with the help of the reverse transcriptase. All this chemical reverse transcriptase just converts this RNA into DNA, just goes and attacks the DNA of a macrophage and just takes control over it. Then afterwards, this viral DNA is just started producing. It almost act like an industry. So place of production of macrophages. This way just act like an HIV factory. A huge amount of HIVs are just produced. After this, the T helper cell just keeps on producing this HIV. T helper cells or T lymphocytes. It just replicate and just produce huge number of HIVs. So from that, new different types of progeny, n number of HIV virus will be produced. So all this time, what happened? B lymphocytes will not go and attack this cells. Why? Because this it's belong to our body. That is T helper cell just belong to our body. This B cells have got no idea that HIV virus has just came, attacked this T helper cell, and just started producing n number of HIV virus. Gradually, this HIV virus keep on multiplying. It just goes and affect the nearby T helper cells and just keep on increasing. It leads to the progressive decrease in number of T helper cells. During this time, what happens is people suffer bolt of fever. Bolt of fever means frequent fever just comes, and diarrhea, continuous for long days, and weight loss. So why? Because of decrease in number of T helper cells, and also this person again started infecting from lots and lots of other diseases. Why? Because your immune system has been reduced. It just keeps on affected by lots and lots of other diseases. So what is a simple blood test? So what are the blood tests can be done is, it can be done for ELISA. So ELISA test is just helpful in finding the HIV antibiotics in the body. But the best 100% confirmatory test is the Western blot test. So with the result of this Western blot test, you can have a 100% confirmation whether that particular person is HIV positive or negative.